What's going on, guys? Welcome back to another Spelunker Neuroglancer guide video where today we're going to be talking about using colored annotation point pairs. So we did a point pair paths guide earlier. This one will be similar to that, so if you've already seen that, this might just be extra information. Where this guide differs from that guide is we are using colored point pairs in this. So when you have more than two paths, or even when you have two, you do have the option of changing the colors of the points. And this can help distinguish the paths for you visually so that you can make a better judgment on whether or not your cell is taking a certain path. So we have a pretty difficult area here. As you can see, this type of merger, we have a tear in the segments, we have a membrane swirl, we have missing slices, and we have some biological merger type things going on as well, which is a merger where the natural membrane is almost invisible, which can be tricky for the AI to pick up, and it's more of a human-oriented task to be able to recognize those, at least for now. So let's take a look at what we have. We have this red cell, and it looks like, let's take a look at the segmentation here. It looks like the red cell and so if you hit the star, you can make it disappear. That's not what I wanted to do, but we know where it is. If you hit the eyeball, you can make it disappear, but not off the list forever. And we can hit the eyeball here, we can hit the eyeball there, and we can hit the eyeball here. So that's what the trails look like without those cells on. As you can see, they make pretty nice paths. But when we look at it all together, it's a little bit more difficult to tell, especially this red one. Like that's pretty difficult to tell there, right? So there's clearly some missing data, or the data is merged with the other cells. As you can see, this orange piece maybe has a piece of that red piece, and this pink piece also has a connection to this red piece. So let's take a look. Let's get into it. Let's see what's going on here. And let's see where these point paths lead. So this type of finding the paths can be difficult. So what you're going to want to do, as in the previous video, is you start further away from the sort of accident area. And you just go slice by slice. Say we're doing the yellow one first. That's pretty difficult to see, but this sort of shape you keep in your head, you go through one terrible slide, that shape is still kind of here. And you can instead of following it through the torn area, you can follow it through the larger area, more seeable area, thicker area, instead of this thin area, which would be a little bit difficult. But sometimes if you go through the slide, be like, okay, this is the same as this, so I know that if this connects to this, that is probably also the same. So now we followed that through that first error, and now we're getting to this connection here. So if we scroll in, and let's see which, so yellow one is the fifth annotation. If we turn that off, we can see that this orange connects to this red. You could see the ER, whoops, turning things off. This endoplasmic reticulum is going in to this cell, and it's still here. So that is a pretty solid connection. So we could say, okay, this, let's see which, is it over here? Where'd the point pair go? There it is. Okay, so that point pair, the yellow path is good. So this connects to that. So that's good to know. Now that we know that, we can keep on scrolling. And now let's see where this path goes. So one thing you're going to want to do, as you can see, the red path that we just connected to the orange path is merged with this purple path. So what we did is we, in these types of areas, what you're going to want to do is usually click on a bunch of stuff, just see what's there, see what looks like it's going the same direction. Like this doesn't look like it's going. This teal one looks like maybe it's going in a similar direction, but it's a little bit askew. This purple one seems fine, and it's pretty intact even through the errors, so that's nice. But this one, it's right in the membrane swirl, so we're going to have to solve that one with the blue path. So once again, you start a little bit further away. This slice is pretty valuable because, let's turn off this point pair. You can almost see the entire path that like this connects to that. And like we said earlier with the yellow one, using the larger, more intact areas as a guide visually can help you draw the path through an error, especially in these parallel plane mergers and damage slides. So let's turn the segmentation back on. So as you can see, that this path is a little bit skew, so it'd probably be more like that. But since the annotation points are just a guide, they don't need to be exactly perfect. And as you can see, it's intact over here, so we know this is good. So even though that this swirl kind of breaks these pieces and causes a merger, we know that this is the correct path for this red piece to this purple piece. So what you would do probably mid-path making is you would want to split as you go so that you have more colors instead of one solid color that is connected to multiple paths. So there's that path solved. So now we know that the orange connects to the red via the yellow path. We know that the red connects to the purple via the blue path. And there was the third path, right? Where's the green? Here's the green. So this red we know is merged because it connects to two different things. So had we been splitting along the way, this would make a little bit more sense. But because we haven't been for training purposes, we can follow the red through even to a third path. And that helps us to make sense of the merger thus far, because the red splitting into two things is already crazy, but splitting it into three is even crazier. So we solve those paths with 3 different point pair paths, 3 different colors, and by using these color-coded paths, we are able to create a way to solve these troubled, damaged areas in a systematic way. And thus, using these techniques, you can yourself solve tough areas that seem very difficult to get through if you try to keep all the locations in your head without using point pairs and without using color coding. So hope this one helped you, and I will see you guys in the next one. Take it easy.